Hello everyone, you're welcome on my YouTube channel Capsule. This is the trailing video of my previous video on biopharmaceutics consideration in dosage form designing. I, Dr. Vinay Kumar Rai. In this video, I'll tell you about the factors affecting drug absorption and bioavailability. So, what are the different factors that affect the absorption rate, extent of absorption, and bioavailability? The first factor is pharmaceutical factor, that include the factor relating to the physicochemical properties of the drug as well as the dosage form. So here we will firstly consider the physicochemical property of the drug substance. So as far as the drug substance is concerned, different physicochemical properties and the other properties that affect the rate or extent of absorption are the drug solubility and dissolution rate. As you all know that absolute or intrinsic solubility is defined as the maximum amount of solute dissolved in a given solvent under standard condition of temperature, pressure and pH, and it is a static property. However, the dissolution rate is defined as the amount of solid substance that goes into solution per unit time under standard condition of temperature, pressure and other humidity, other environmental condition. So it is considered as a dynamic process, and there are several drugs that have poor aqueous solubility that affects the dissolution rate. So the matter is of great concern when the solubility is less than 1 to 2 mg per ml in the pH range of 2 to 8, that is the usual pH of range of gastrointestinal tract, and this will severely affect the dissolution rate of the drug. However, the drugs like C-saccharide shows good bioavailability despite their low aqueous solubility, and that is the exception. And two reasons can be attributed to this. Because of this exceptional reason, the first is the rapid rate of dissolution despite low intrinsic solubility of this drug, and the second one is the therapeutic dose of the drug may be so small that the GI transit time is sufficient for adequate dissolution and absorption to occur. So ultimately the dose is very low, and the bulk or amount of the GI tract which is available for the dissolution of this particular drug is very much high. So thus in contrast to absolute solubility, the dynamic process of the drug dissolution is better related to drug absorption and bioavailability. So this is how it affects the drug absorption rate and extent. Now the second one is the particle size and effective surface area. As we all know that the particle size plays a very important role in increasing or decreasing the solubility of the drug, and particle size and surface area of the solid drug are inversely related to each other. So smaller the drug particle size, greater the surface area, as you all know, and two types of surface areas are there. So absolute surface area and effective surface area. So absolute surface area, which is the total area that solid surface of any particle contain, and the effective surface area, which means it is the area of solid surface exposed to the dissolution medium. So according to the modified Noyes-Whitney equation that explains, the larger the surface area, the higher the dissolution rate will be. Since the surface area increases with the decrease in particle size, so it will ultimately lead to the higher dissolution rate, and the particle size reduction can be done with the help of micronization technique. Now the next point is polymorphism, and it is also called amorphism. So depending upon the internal structure of the drug, a solid can exist in crystalline and amorphous form. So when a substance exists in more than one form, the different forms may have a different physicochemical properties, and that is why we always say that amorphous form of the drug has greater solubility than the crystalline form of the same drug. So for example, The amorphous form of Novobiocin is 10 times more soluble than the crystalline form of the same drug. Here, the order of the dissolution of different solid forms of the drug is amorphous, very high, then metastable form, somewhat between amorphous and crystalline form, and crystalline is somewhat stable, but it has lesser solubility than the other forms. Now, the fourth point is pseudopolymorphism, in which, as we all know, that the crystalline form of the drug can either be a polymorph or a molecular abduct or both. The stoichiometric type of abduct, where the solvent molecules are incorporated in the crystal lattice of the solid, are called as solvates, and the solvates can exist in different crystalline form called as pseudo-polymorphs. When the solvent in association with the drug is water, the solvent is known as hydrates, and generally the anhydrous form of the drug has greater solubility than the hydrates. So this is because the hydrates are already in interaction with the water and therefore has less energy for crystal breakup in comparison to the anhydrate. That is why these have a greater solubility than the hydrates. Now, the next one is the salt form of the drug, where the most of the drugs are either weak acid or weak bases, as we all know one of the easiest approaches to enhance the solubility and dissolution rate of such drugs is to convert them into the salt form. So generally at given pH, the solubility of drug, whether acidic or basic, its salt form is constant, and the dissolution rate of weak acid gets increased with GI pH, and vice versa is also true. So this is how this particular parameter affects the dissolution or drug absorption rate and bioavailability. Now, the lipophilicity of the drug, it significantly affects the drug absorption rate and action, and ultimately bioavailability also. Since most of the drugs are weak electrolytes, their degree of ionization depends on the pH of the biological fluid. So if the pH on either side of the membrane is different, then the compartment whose pH favors the greater ionization of the drug will contain greater amount of the drug. So, and only the unionized or undissociated fraction of the drug will be absorbed easily if sufficiently lipid soluble. If the unionized form is sufficiently lipophilic, so it will absorb easily and can permeate the membrane passively until the concentration of unionized drug will be same on either side of the membrane. Now, the next point is the pK value of the drug and gastrointestinal pH. As mentioned before, it is the pK of a drug that determines the degree of ionization at the particular pH, and that only the unionized drug, if sufficiently lipid soluble, is absorbed into the systemic circulation. Thus, even if the drug exists in the unionized form, it will be poorly absorbed if it has poor lipid solubility. So lipid solubility is very important terminology that governs the rate of drug absorption into the systemic circulation. Now, three major drug characteristics that majorly determine the passive transport or permeability of the drug across the intestinal epithelium are lipophilicity of the drug, polarity of the drug, and molecular size. Yes, you hear that right. The molecular size is very important and crucial because upon increasing the molecular size of the drug, the absorption rate may differ or may reduce. So it is very important. Now the next point is drug stability. Drug stability is very important point, and a drug for oral use, as you all know, that it may destabilize either during its shelf life or in the GI tract. Here, we are talking about the stability of the drug inside the gastrointestinal fluid. Now, two major stability problems can be observed uh, that may result the poor bioavailability of an orally administered drug, that is degradation of the drug into the gastric fluid and interaction with of the drug with a food product or other drugs that may affect its absorption into the systemic circulation. So it is also a very important parameter that can be considered. Now, next point is stereochemical nature of the drug. It is a well-known fact that chiral drugs constitute approximately 60% of the drug in current use, and majority of these are marketed as racemic mixture. Although it is a well established that two optical isomers differ in their potency of pharmacological effect. So enantiomers possesses identical physical and chemical properties despite significant differences in spatial configuration. The biological processes which are passive in nature do not display selectivity for one isomer. However, biological processes such as protein binding, which require interaction of drug with a macromolecule, may exhibit stereoselectivity. Today, the majority of drugs are absorbed passively and they do not display stereoselectivity. So it can be of minor importance, but it is considered while studying the rate of drug absorption and the effect of different parameters on the effect on the drug absorption rate and action. So after studying the pharmaceutical factors related with the drug substance, now we will see here the dosage form related factors, pharmaceutical factors. And first one is the disintegration time and dissolution time of the drug. The disintegration and dissolution time is of particular importance in case of solid dosage form like tablets and capsules. So in vitro disintegration test is not a guarantee of drugs viability, because if the drug is getting released easily from the dosage form, that doesn't guarantee the drug is being available inside the systemic circulation. It has to be permeate through the biological membrane. Now, in vitro disintegration test is not a guarantee of drug's viability, because if the disintegrated drug particles do not dissolve, then it will not be absorbed. So, however, if a solid dosage form does not conform to the DT, it presents viability problems because the subsequent process of dissolution will be much slower and absorption may be insufficient. Just like in the case of coated tablets, especially sugar coated ones have a longer DT than normal tablets. So rapid disintegration is thus important in the therapeutic success of dosage form is partially true. So dissolution rate is directly related with the drug absorption. Slow dissolution leads to the slower bioavailability and vice versa. Now, the second point is manufacturing variables. Here, different manufacturing variables can affect the drug absorption process, and the drug dissolution is the single most important factor in the absorption of drugs, especially from the most widely used conventional solid dosage form, tablet and capsule. So, the three parameters: method of granulation, compression force, and intensity of packing. All these parameters are important. Now, pharmaceutical ingredient or excipient related factors. Here is the third point, that is pharmaceutical ingredient or excipient. Several pharmaceutical excipient or ingredient are used to develop a tablet or capsule, and these can be vehicle, diluent, binder, granulating agent, disintegrant, lubricant, coating agent, suspending agent, surfactants, buffer, complexing agent, colorant, precipitation, crystal growth inhibitors, and so on. So all these ingredients are used to develop product. So the presence or absence of these ingredients may affect the dissolution process of the drug, and ultimately it will affect the absorption of the drug. Now, the next point is nature and types of dosage form. Yes, it is also important. As a general rule, the bioavailability of a drug from various dosage forms decrease in the following order. First one is the solution, then emulsion, following suspension, capsule, tablet, and coated tablet, then intermediate coated tablet, and sustained release products. So the drug dissolution process decrease in this order. Now the next point is product age and storage condition. It is also very important based on the duration of the storage and based on the condition in which the drug has been stored. It also affects the absorption process because some of the physicochemical properties may change with the time. Now, after studying these factors, now we will see the patient-related factors that can affect the rate of drug absorption, and patient-related factors may include the factors related with the anatomy, physiology, and physiological characteristics of the patient. Now, the first factor is age. In infant, the gastric pH is high and the intestinal surface and blood flow to the GI is low, resulting in altered absorption pattern in comparison to the adults. However, in elderly person, that causes an impaired drug absorption include altered gastric emptying, decreased intestinal surface area, and GI blood flow. There will be the higher incidence of achlorhydria and bacterial overgrowth in small intestine. So these are the age-related factors that can affect the absorption process. Now, the next point is gastric emptying time. And yes, gastric emptying time sometimes can be delayed and can be faster. So apart from the dissolution of drug and its permeation through the biomembrane, the passage from the stomach to the small intestine called as gastric emptying, and can also be a rate limiting step in drug absorption because these are the major site of drug absorption is intestine, and if the gastric emptying is delayed, then the rate of absorption will be delayed and the bioavailability will be less in that condition. A large number of factors that influence the gastric emptying time may include the volume of the meal, based on the volume you are consuming as a meal, and composition of the meal, whether you are taking fat, proteins, or carbohydrate, physical state or viscosity of the meal, whether you are taking a solid food or liquid product, temperature of the meal, either you are taking meal in hot condition or in cold condition, and gastrointestinal pH, electrolyte and osmotic pressure, body posture, you are sleeping or you're just sitting around, and emotional state, you are happy or you are in stress condition, you are doing exercise or your body is in normal position, disease state, and whether you have taken some drugs or not. All these factors may affect the gastric emptying time, and ultimately these factors will alter the rate of the absorption of the drug. Now, next point is intestinal transit time. So intestinal transit time is also very important. The residence time that depends the intestinal motility or contraction. So the mixing movement of the intestine that occur due to peristalsis contraction promote drug absorption fastly by increasing the drug intestinal membrane contact, and secondly by enhancing the drug dissolution, especially of poorly soluble drugs through induced agitation. So peristalsis movement of the drug can also affect the rate of the drug absorption. Now gastrointestinal pH is the next point. Tremendous, that is 10 to the power 7 folds difference in the hydrogen ion concentration is observed between the gastric and colon fluid. So the GI pH generally increases gradually as one move down to stomach to the colon and rectum. Now, GI fluid pH influence the drug absorption in several ways. So, we will study these factors in the next video in detail. Here, I am just taking the general factors that can affect the drug absorption or bioavailability. Now, disease state. Obviously, if you are in disease condition, then your body temperature or peristalsis movement may alter, and those conditions may lead to the altered absorption rate and extent. So several disease states can influence the rate and absorption of the drug. The three major classes of disease state may include gastrointestinal disease, cardiovascular disease, and hepatic disease. All these factors may affect the rate of the drug absorption. Now, blood flow through the GI tract. The GIT is extensively supplied by the blood capillaries network. So, if the blood flow around the site of the absorption is high, then a rate of absorption will be high, and vice versa is also true. So this is how the rate of the drug absorption get affected by blood flow around the GI tract. Now the next point is gastrointestinal content. Whatever the content is present inside the stomach or intestine, that will ultimately alter the rate of the absorption of the drug. It may be food, fluid, or the normal GI content. There may be possibility of food-food interaction, fluid interaction, drug-drug interaction. So all these conditions may lead to the altered absorption rate. Now the next point is pre-systemic metabolism. Yes, it is observed by the liver, and there are several enzymes present on the GI tract and your intestinal cavity. And these enzymes are luminal enzyme, gut wall enzyme, bacterial enzyme, hepatic enzymes, and all these enzymes are responsible for the metabolism of the drug and may alter the rate of the absorption of the drug. So this is all about this topic, and we have included all the factors that may affect the absorption of the drug, and if the absorption of the drug is affected by these factors, then ultimately bioavailability will be reduced. So this is all about this video, and in my next video I'll tell you about the pharmaceutical factors affecting drug absorption. That is part 1. So keep watching us. Thanks for watching.